30s I did nuclear fusion research, and it was great fun. There were really challenging physics problems there. Eventually I decided that I needed to do something a bit more practical, and from my experience in fusion, I had used supercomputers a lot at Lawrence Livermore Laboratory, and I helped Sid Caron write a proposal to found the Supercomputer Center here in San Diego. I stepped down from management, and in recent years I've been doing bioinformatics and, you know, having another career and having a great time. There's truly a flood of data coming from these sequencers. One way to appreciate that is that a state-of-the-art sequencer like the Illumina HiSeq 2000 can generate about 100 gigabytes of data each day. There's probably the equivalent of 1,000 such machines installed, which is of the order of 100 terabytes of data each day. And, you know, over a year, that's tens of petabytes of data. So we're talking about a lot of data. That's just the raw data, but in the process of analyzing the data, there's several times as much. The sequencers don't give you a complete genome. The capability of sequencers at the current state is such that they can only output you know, typically of the order of 100 bases at a time. So the human genome has 3 billion bases roughly, and that amount of DNA gets cut up into many small pieces and called reads, and the read length for the Illumina sequencer right now is of the order of 100 bases. So a researcher needs to take all these short reads and either map them against some reference genome or try to reassemble them into a full genome. It's like taking the pages of a book and cutting them up into small pieces and then trying to reassemble the book from that. And you can't just take one copy of the book and reconstruct it all, because there are errors in the data, and so you have to have many copies of the book. So that leads to a significant increase in the amount of data. So you ask what this is going to look like in 10 years. I don't even know what it's going to look like in 2 years from now. There are major developments in the sequencing technology that could really turn things around. The sequencers right now, like the Illumina sequencers, produce reads that are 100 bases long. But there's technology in the offing that could increase that many-fold. That will, on the one hand, simplify analyses, but on the other hand, it will mean that many of the codes will need to be changed and different kinds of algorithms will be required. Although the cost of storage and computing is coming down, the cost of a person to do the analysis is not on Moore's law. This is important in personalized medicine because although that cost will gradually come down as better tools are developed, it's not coming down at anywhere near the rate at which the sequencing cost is coming down. It's clear there's tremendous potential here, and there's no doubt that this technology will be adopted for personalized medicine, but there are all manner of challenges there. Not the least of which is training doctors to interpret the data. So sophisticated kinds of tools will need to be developed to help if personalized medicine is really going to realize all the benefits that people anticipate. A typical kind of analysis would be to look for differences between the subject genome and some reference genome. And the simplest differences are called SNPs, single nucleotide polymorphisms, where there's just one base, one DNA base in A, C, T, or G, which is different in the subject from the reference. And the effect of that difference could be negligible or it could be very serious. Those kind of differences can be found by doing alignments, or they can be found by doing these de novo assemblies. The analyses of the data right now generally require a fair amount of skill in computing, and this is a bit of an obstacle for many biologists. For phylogenetic analyses at SDSC, we've produced a portal or a gateway called the Cypress Gateway, which simplifies the analysis for typical biologists, so they don't need to know about Linux or job schedulers or parallel coding. That's all screened from them through a web browser interface that we've developed. And that's been extremely successful. We have thousands of users around the world who've used this browser interface to do phylogenetic analysis.